Hot Start PCR is a method of DNA amplification. It is a modified form of the traditional polymerase chain reaction method. However, this technique reduces the production of non-specific DNA. It is also a convenient method to follow as it can be set up at normal room temperature and does not require special optimization in this regard. Potential of traditional PCR was acknowledged. Scientists pivoted towards the modification of the original technique in order to achieve maximum yield with enough efficacy. The results of their research led them to Hot PCR. Mechanism of Hot Start PCR. It works on the same mechanism just like normal PCR by using DNA polymerase enzyme for synthesizing new strands of DNA from a single strand of template DNA, but with a few advancements. These modifications involve the usage of excessive heat and the methods of separation. The separation method includes alterations in Taq polymerase activity by inactivating its binding when required or adding it late into the sample. It leads to a higher yield with increased specificity. Prevention of non-specific binding. When the reaction mixture is completed after denaturation, then it reduces the chances of non-specific binding of primers, priming, or the development of primer dimers. The methods which can be adapted to complete the reaction mixture at such high temperatures include certain modifications and alterations which could prevent the activity of DNA polymerase at a low temperature range. Other than that, modification of dNTPs or adding an essential reagent after denaturation has taken place would give the required results. Applications of Hot Start PCR. This method has many applications in the medical and industrial fields. The examples include the usage of PCR in forensics where DNA fingerprinting techniques are used. Their amplification of DNA is required to catch criminals, testing to detect parents, cloning, and detection of any mutation, genetic testing, biodefense, and sequencing of DNA. Reduction in non-specific amplification. When the temperatures are low, the amplification of DNA leads to non-specific DNA production. But, Hot Start PCR reduces this amplification, which was initially a major problem in the normal PCR method, but it got eliminated by this modification. The alterations in this method make sure that the enzymes which are present in the mixture are not active until the normal binding temperature is attained. When the production of non-specific products is inhibited at the start, the overall specificity of the PCR process is substantially increased. This feature makes it the most important component of diagnostic procedures.